Okay, folks, let's get going. We have two fabulous talks today from the Cancer Center. Going back to our format of having two 23-minute presentations with 7 minutes for questions. The first speaker is James Hansen. Dr. Hansen is an assistant professor of therapeutic radiology who will be sharing with us some of the most exciting translational work that has been done at Yale, work that he's done along with Dr. Peter Glaser on harnessing a lupus autoantibody for cancer therapy. Then following that, we'll hear from Dr. Xavier Lohr. Dr. Lohr is the co-director of the co-director of the cancer genetics program at Smilo. He's a gastroenterologist who studies hereditary colon cancer, and Dr. Lohr will be talking to us about hereditary colon cancer syndromes, achievements, and challenges. Dr. Hansen.
Great. Well, thank you very much for that introduction, and thank you to everybody for coming here. I'm James Hansen, assistant professor in radiation oncology, and today I'm very excited to talk with you and give you a little glimpse of some of the work that we've been doing, trying to figure out if there actually is a way to harness a naturally occurring lupus antibody and maybe actually apply it towards cancer therapy. Now, for those of you who are wondering what's up with the purple butterflies, the butterfly, of course, is the symbol of lupus, and we're trying to show these butterflies representing antibodies and their interactions with DNA here. As to what exactly they're doing with the DNA, well, that's what we're about to talk about. I don't have any relevant disclosures to make. So just to jump right into it, I'm sure that all of us in this room would agree, key goal is to find therapies that are selectively toxic to cancer cells and leave normal cells alone. And when we think about all the biomolecules available to us that we could potentially use in that regard, I think monoclonal antibodies have to stand out as some of the most compelling candidates. And that's because they're so incredibly specific in their binding for their target antigens. But of course, this is not a new idea. Antibodies have been applied to cancer therapy for years, and with some great success. But one important point to recognize is that all the antibodies currently approved for cancer therapy are binding to things that are outside of the cell, like growth factors or receptors. And that's because the main dogma related to antibodies for years has been antibodies do not penetrate cells. They can't get inside the cell. And that leaves a whole host of potential tumor targets inside the cells inaccessible to this approach. And that's been a limiting factor in antibody-based cancer therapy for years. But now, believe it or not, we may just find a solution from a disease called lupus. Lupus is the classic prototypical autoimmune disease. It's characterized by widespread inflammation and tissue destruction as the patient's own immune systems mount an attack against their own tissues. Now, this is a very mysterious disease, and one of the classic hallmark laboratory findings associated with lupus is the presence of circulating antibodies reactive against the patient's own DNA. Now, these anti-DNA antibodies are also very mysterious. They were first recognized as far back as 1957, but despite now more than 50 years of research, we still don't know exactly why they're made, and we still don't know exactly how they contribute to lupus pathophysiology. What we have learned, however, is that a rare subset of these anti-DNA antibodies actually has the unusual ability to cross through membranes and get into the nucleus of living cells. Not all these antibodies do this, but some do. So let me introduce you to the hero of today's story, the antibody called 3E10. 3E10 is actually a mouse antibody. It's isolated from a lupus mouse model. It binds DNA, and it is remarkably effective in penetrating into the nucleus of just about every cell that it comes into contact with. Now, what we're showing here are some confocal microscopy images zoomed in on the nucleus of cells that have been treated with 3E10. In this case, 3E10 has been tagged with a FTSE tag, so it shows up in green. The DNA is in blue, and you can clearly see that when you overlay the two, not only is 3E10 getting into the nucleus, but it's associating with DNA. Now, what's even more remarkable about this antibody is not only does it get into the nucleus, there's a lot of antibodies that do that, but this antibody does so in a way that's not toxic to normal cells or tissues. So when you start thinking about having an antibody that can get in the nucleus, doesn't do anything bad to cells, you have to start thinking there must be some cool things we can do with this in terms of molecular therapy approaches. But before we get too excited about this, let's maybe start answering some basic questions that I'm sure are starting to cross your mind. Number one being, how on earth does this happen? Antibodies do not do this. They don't penetrate cells. So what's so special about 3E10? Well, I can give you some answers. So let's take a quick look and remind ourselves of the basic structure of an antibody. 2 heavy chains, 2 light chains, and each chain composed of a variable region and a constant region. Now, some antibodies that penetrate cells do so via their Fc tails via interactions with Fc receptors on cells. That is not the case for 3E10. If you take 3E10 and you construct from it what we call a single chain variable fragment, which is composed only of the variable regions, the light and the heavy chains, and you apply that to cells, you find then that penetrates into nuclei just as well or even better than the original antibody. So that tells us that whatever it is that's magic about this antibody, whatever allows it to penetrate into the nucleus, is contained in the variable sequence, has no requirement for the constant regions whatsoever. Now, this is about as far as you can go in terms of minimizing the structure of 3E10. If you just take the variable region of the light chain, that will not penetrate. Just the variable region of the heavy chain, that will not penetrate. The two have to be together. And that's therefore implying that the actual antigen binding site is somehow critical to this function. Now, I could spend another 3 hours explaining how we got to the next slide, but just to jump to the punchline, it appears that the antibody is actually penetrating nuclei through a specific nucleoside transporter called ENT2. It took us a long time to get here. I spent a large part of my postdoc working on this, actually. Nothing seemed to be able to successfully block the ability of the antibody to penetrate cells until we found that certain mutations that prevent it from binding DNA also prevent the antibody from getting into the nuclei. So that led us to start thinking about mechanisms of DNA uptake, nucleoside salvage pathways, and eventually we found the antibody cannot penetrate into cells if this one transporter, ENT2, is not there. If you take those same exact cells and you put that transporter back in, ENT2, suddenly you've restored the ability of the antibody to penetrate back into those nuclei. So what we are now proposing as our model is that the way the antibody penetrates the nucleus, as shown in this artistic depiction here, is that it binds to DNA or to nucleosides outside of the cell, and then thereby activates and accesses that ENT2 pathway and gets into the nucleus. Now, that's a very unusual way for any proteins to get into a cell. And what we actually like about this is that we believe this then means that anything that you carry into the cell with the antibody should be able to avoid lysosomes and not be targeted to endosomes and such. So whatever you carry in hopefully has a better chance of retaining biological activity. But of course, this is all based on cell culture work. If we're really going to think about using this as a therapeutic approach, we need to know where it goes in vivo. And interestingly, when you administer the single chain fragment to mice with tumors or mice with certain tissue damage models, you see a very striking pattern. And that is that the antibody really does not go to the normal tissues at all. It does localize quite well into tumors. And in a rat stroke model, it actually localizes specifically into the area of the infarct. Now, we are still working out the precise mechanisms of how exactly this works, but we're finding now that the presence of extracellular DNA actually facilitates and enhances uptake by the antibody. And so the working hypothesis here is that in vivo, the antibody is attracted and penetrates best into tissues where there's going to be a higher concentration of extracellular DNA, such as you would find in areas of ischemia in a stroke or in areas of necrotic tumor that are releasing their DNA. Regardless, it looks like this antibody has good potential as a delivery agent to sites like tumors or sites of damage. And we actually have done some work in this regard. We've actually made quite a few fusion proteins to test this, and I only have time to tell you about one, and I'm choosing this one because it helps us transition to the main point here. We wanted to use the antibody to carry a heat shock protein into cells, with the idea being that this heat shock protein would make cells happier and able to resist things like oxidative stress in the settings of stroke and myocardial infarction. So we made it an FV HSP70 fusion protein, and we showed that it was able to penetrate into the nucleus of neurons in culture quite well. And then remarkably, when applied to this rat stroke model, not only did we find that it gets into the area of the infarct, but it actually exerts an improvement of an effect. It actually reduces the size of the infarct and reduces the actual neurologic outcome in terms of complications after the stroke. So pretty impressive, biologically active. It gets into the region of the stroke as well. And as I said, a lot of this work was what I had done at UCLA prior to coming to Yale. By the time I came to Yale, I was still fascinated by this antibody, and I wanted to continue working with it here. And I came up with the idea that I'd like to use this FV-HSP70 protein as a biodefense mechanism. I figured it would protect cells against the effects of ionizing radiation. And so I quickly tested this hypothesis, as shown here, and this is where everything gets turned upside down. What we found was, indeed, actually, the FV-HSP70 does provide a degree of radioprotection, but the big surprise was the control arm. The antibody fragment by itself actually makes the cancer cells more sensitive to the effects of radiation. Now remember, this was not supposed to happen. The antibody is not supposed to do anything bad to cells at all. But it turned out to be quite reproducible, not only in this cell line, but in multiple cell lines. These are some U251 glioma cells, and I think the take-home point from this slide here is the antibody fragment by itself is not toxic to the cells, but in the combination with radiation, we see a synergistic effect. And this happens in vivo as well. These are some nude mice bearing glioma xenografts. Again, the antibody by itself does nothing to the tumors, but in the presence of the antibody, the effect of radiation is amplified. So we've got a new phenomenon here that's reproducible in cell cultures, it's reproducible in an animal model, and a little bit of luck came my way at this point because I just happened to be doing this work in collaboration with Dr. Glaser, who's of course expert in DNA repair. And then in following collaborations with virtually everybody in our departments, we were able to actually quickly get to this slide here, which summarizes what we found. In fact, it's probably not that big of a surprise now to think about it, but it surprised us back then. 3E10, being an anti-DNA antibody, turns out to preferentially bind to DNA substrates with free single-strand tails, and in doing so, it actually impairs mechanisms of DNA single-strand and double-strand break repair. So not only did that give us a mechanism as to how this antibody is radiosensitizing and chemosensitizing tumors, it also allowed us to start making some other predictions as to how can we use this antibody as a therapy by itself. And this invokes the idea of synthetic lethality, whereby certain cancer cells that have pre-existing defects in DNA repair, such as due to BRCA1 or 2 mutations or PTEN deficiency, are very vulnerable to further suppression of DNA repair mechanisms, whereas normal cells can survive this. So we got ourselves a matched pair of human ovarian cancer cells that are BRCA proficient or deficient and treated them with the antibody fragment by itself. No radiation, no chemotherapy. And as we predicted, the antibody fragment was selectively toxic to those BRCA deficient cells. So this really opened up the potential and the idea, maybe we can use this antibody as a selectively targeted therapy for cancers that have defects in homologous recombination, which is conceptually very interesting and I think very exciting. But of course, thinking clinically, if we look at this, we're talking about reducing the survivin fraction down to only about 60%. And I don't see that that's going to be curing any tumors here. So if we want to make this a clinically relevant discovery and therapy, we've still got a ways to go. And that actually is about what I've been working on since I joined the faculty after completing residency. And I've been very lucky to have a great team join me with a postdoc, Philip Noble, and a resident, Melissa Young, and another medical oncology fellow, Jamin Patel, all helping to make this happen. What we're trying to do is we're trying to find ways to optimize this effect. And by optimize, I mean make it more potent and also make it safer. So looking at our starting points, we've got the full 3E10 antibody, and we've got the single chain fragment. So which are we going to choose to work with to make this technology better? Well, remember the full antibody has the Fc tail sticking around. And remember, this is an anti-DNA antibody, which is inherently a dangerous molecule. Lupus anti-DNA antibodies are associated with nephritis. And the way this works is these antibodies encounter DNA in the circulation. They form these immune complexes, which get stuck in the kidney. And then wherever these things get stuck, these Fc tails are pointed outward and can activate things like complement and ADCC and really spell disaster for whatever organ that's attached to. We don't need the Fc tail. It's not required for penetration. It's not required for inhibition of DNA repair. So let's lose it. Let's just keep using this single chain variable fragment. In fact, another advantage of the fragment is it does already carry a mutation in it, D31N, that significantly enhances its binding for DNA. And yet, it's still not very potent. We're talking about dropping the surviving fraction down to, 70% here. We'd like to do better than that. So how are we going to ramp up this effect? Well, with 3E10, it does seem like everything is related to how well it binds to DNA. It binds DNA, it gets into cells. It binds DNA, it inhibits DNA repair. So my hypothesis here is that if we can ramp up this effect in terms of DNA binding, then hopefully we can ramp up its effect on cancer cells at the same time. Now, we've already worked on the binding affinity in terms of that mutation, but there's something yet left to be optimized, and that's the binding avidity. And binding avidity refers to the overall strength of the interaction between an antibody and its target antigen. And it takes into account the exponential improvements afforded by the presence of multiple binding sites. The classic example that I like to use is if somebody was to toss me a baseball and tell me I have to catch it with one hand, I probably won't do it, to be honest. There's a reason I'm not on the softball team. But if you tell me I can use two hands, I probably will be able to catch it. And it's the same thing with an antibody. Give it one hand and it can do its job, but give it two hands and it can potentially move mountains for you. So we've made an easy construct here, simply doubling the number of DNA binding sites, making a divalent single-chain fragment, still with these mutations. We make it in Pichia pastoris. Thankfully, it's easy to make and purify. Well, it's easy for me to make it and purify because I don't do it. Philip does it, and he does a great job of it. It penetrates nuclei quite well, just as well or even better than the single-chain fragment. And the good news is when you put it on the cells that we're interested in, it does indeed have a greater impactive effect. But still is not toxic to the BRCA2 proficient counterpart. So we're moving in the right direction. And this is true in multiple types of BRCA-deficient cells. These are some CAPN1 pancreatic cells. Again, the lower curve is the divalent fragment, and it's clearly more potent than the single chain. Even more excitingly, I think, Phil and I have been testing this construct on a wide range of cells, and we've been seeing that it's clearly not toxic to most cells at all, which it shouldn't be. But we did sort of stumble onto this finding, which is that U251 cells, for whatever reason, turn out to be remarkably sensitive to the divalent fragment, whereas the single-chain fragment doesn't even touch them. Now, didn't expect that, but U251 cells are PTEN deficient, and the association between PTEN status and homologous recombination is a little controversial, but it's clearly there. And if we get the matched pair of cells that have PTEN function restored, we see that the cells have lost their sensitivity to the divalent fragment. So pretty solid evidence that there is clearly an association here between PTEN status and sensitivity to this treatment approach. And one of the reasons I'm very excited about this is that this significantly increases the number of tumors that could potentially be amenable to 3E10-based therapy. BRCA2 mutations occur, but nowhere near as commonly as PTEN deficiency. Just to make sure that we understand exactly what's going on here, we're believing that the reason that the fragment is toxic to these PTEN deficient cells is because they're leading to inhibition of DNA repair, causing accumulation of DNA double-strand breaks. And so we check this by staining for phospho53BP1 foci in the PTEN proficient and deficient cells. And I think it's pretty clear there's a difference there. Lots of double-strand breaks getting induced and persisting in the presence of the fragment in the PTEN deficient cells, but not so in the PTEN proficient cells, which is consistent with what we believe this antibody is doing in terms of inhibiting DNA repair. And this happens in BRCA2 deficient cancer cells as well. So by this point, Phil and I were pretty convinced that we had an optimized version of 3E10 that was worth taking in vivo now. So we went with the CAPN1 model, subcutaneous xenografts in immunodeficient mice. Once tumors were established, treated by IP injection. And I think it's pretty clear that we are seeing a definite separation in terms of tumor growth curves. And what I like even more is that it looks like the mice are not even aware that anything really is happening. Their weights stay rock solid stable. Their behaviors don't change whatsoever. The only caveat that I want to add to this is that these are immunodeficient mice. We still need to run some studies in immunocompetent mice to really see what toxic effects might occur in the setting of an immune system. However, given that these antibodies lack the FC region, I'm still confident that they will be very safe. But still, I would like to see a bigger separation. So we still have some room to improve here. Now, I think this next decision we made is not the world's most incredibly, well, what's the word I'm looking for? We'll just tell you what it is. It's pretty obvious, actually. If 2 is better than 1, 3 is probably better than 2. So we made a trivalent fragment, and there they are, 3 binding sites, and it's a bigger molecule. And this is easy to make too, right, Phil? Almost. Well, the yeast do make it. We can purify it. It penetrates into nuclei very, very well. And the good news is this is even more potent than the divalent fragment. So we're getting more and more potent effects here. We're moving in the right direction to the point that I'm hopeful that this trivalent fragment is going to be our final product. I don't think we're going to need to move to the 4 and 5 and 6 and 7 range. I think tri is going to turn out to be it. And we are talking very regularly with pharmaceutical companies, and we're hopefully going to be having a partner soon that can help us take this into the next level of development. But just to quickly wrap this up, everything that we've been talking about, of course, already is related to one single antibody, 3E10. And there's more than 100 other lupus antibodies known. And so what I'm trying to convey from this one slide here is two messages. One, 3E10 is a very cool antibody. Get it, right, with the iceberg? Number two, I think 3E10 is just the tip of the iceberg. So many other antibodies yet to be looked at. And that's been another arm of my lab so far. And in fact, with Melissa Young and Phil Noble as well, we started looking at cell-penetrating anti-guanosine antibody. What's really remarkable about this antibody is it penetrates cells very well and seems to go everywhere except the nucleus. And there are some thoughts that it may actually be localizing with mitochondria. But we're still working this out. But what's really interesting, remember this is an anti-guanosine antibody. It turns out that it seems to be perturbing G protein signaling and has a selectively toxic effect on KRAS mutant cancer cells. So this antibody really has my attention now as well. And we're going to be working on this further, and hopefully we'll have some more news in the near future. And finally, just to wrap this up, I think it's also important to ask the question, what does all of this mean for the disease lupus itself? If you actually take a lupus population and you ask what is the overall cancer risk compared to a normal population, there is a slight increase. But if you go cancer by cancer, it turns out that increase is driven heavily by the hematologic malignancies. If you go tumor by tumor, you see a pattern fallout wherein certain tumors occur with lower than expected frequencies. And that includes tumors like breast cancer and ovarian cancer. And in particular, if you really look in depth at the breast cancers, it's only the ER-negative breast cancers that are not occurring in lupus. And of course, ER-negative phenotype and BRCA mutations have a direct association, it seems. So it's very tempting at this point to speculate that the reason that there's lower than expected rates of certain cancers in lupus may be due to these circulating antibodies like 3E10 that are suppressing the development of these tumors. Very speculative at this point, but it's gaining a lot of interest. Got a lot of collaborations with rheumatologists around the world now, and hopefully we'll be able to prove this in the near future. So with that, I'd like to thank you for your attention, and I'm happy to take any questions.